much to Jugu and Farhat for organizing this really interesting workshop and for including me. I think I'm a little bit of an outlier, and then I'm going to focus a lot more on the molecular and cellular scale, maybe slightly smaller data, but I hope I will try to keep you interested in the questions that I'm posing. So we are interested in, oh, all right. Hold on, there we go. We are very interested in non-genetic cell-to-cell heterogeneity. A lot of people in this session have already introduced single-cell data, and I think single-cell data is really captured our imagination because there's so much information contained in it. But I also want to remind you that a lot of systems we think use cell-to-cell heterogeneity as a biologically functional way to handle different environmental cues. So, for example, sporulation in B. subtilis or differentiation in embryonic stem cells, some of these things, non-genetic cell-to-cell heterogeneity, we think underlies some of these important biological functions. And we, in my lab, are really interested in how this might apply to macrophages. So macrophages are cells of the innate immune system, and they exhibit a high degree of plasticity to perform many biological functions. So if you think about wound healing, for example, macrophages first come in and sort of perform a pro-inflammatory sort of pathogen fighting function. And then over time, they will change their functionality to become even more anti-inflammatory and resolving, and then participate in things like matrix remodeling and angiogenesis, and basically to heal the wound. And so we kind of, one of the things that the overarching hypotheses that guides my lab is this idea that maybe macrophages are an interesting model to think about how non-genetic cell-to-cell heterogeneity might be a strategy by which macrophages can flexibly mount these different functional responses. And so I'm going to explore with you two different questions today and two different ways that we are looking at this. And the first is going to focus on this inflammatory response, the microbial defense response, and also how the innate immune system then mounts an inflammatory response that then can participate in education of the adaptive immune system. And we're going to ask this question: how do macrophages ensure specificity and control when activating the inflammatory response? And so we've already heard today a lot about why this question is really important. For example, in COVID-19 patients, the inflammatory response is sort of out of control. And yet, you know, certainly proper inflammatory and innate immune responses help us in responses to infections and vaccines. And so really it's important to understand like how the immune system is controlling this, and macrophages are one of the major cell players. So I just want to first give you a very brief summary of what we already know. We already know a lot about these sort of molecular and cellular level responses here. So we use LPS, the component of gram-negative bacteria, as our stimuli. We add it to cells, and then what happens is macrophages produce TNF, the inflammatory cytokine, interferon beta, an antiviral cytokine, as well as other cytokines and chemokines. But what happens then is that TNF actually can go and bind to TNF receptors on the same cells or neighboring cells, neighboring macrophages. And that actually elicits the production of more TNF as well as more cytokines and chemokines. So that's a strong positive feedback loop. And then we also know that TNF as well as interferon beta can bind to receptors on the cell and cause secretion of interleukin-10, which is a negative regulator. It's an anti-inflammatory cytokine, and it's a strong negative regulator of the immune response. So there's 2 points I want to make by going through this. One is that the LPS response is actually a very complex cytokine cascade, right? So we think of the LPS response as being the sort of one thing, but it's actually an interaction of many positive and negative feedback loops. And then we might ask ourselves, well, if all these cells are producing things kind of variably, how does cell-to-cell variability in secretion, in this case, affect this cascade? So this is a question that we were really interested in part because my lab uses a technology, an antibody-barcoded microwell array, in which we can measure secretion from single cells. So in this technology, cells are isolated in these wells, and we are able to measure about 12 to 15 cytokines or chemokines that the cell produces. When they're sort of isolated in these wells, they're stimulated and they secrete. What's important to remember here, so I should say 2 things. One, we use this is a great profiling technology. It's wonderfully complementary data to single cell RNA sequencing. We have used it to profile immune cells in tumors and wound healing. But here I'm going to show you how we're using it to look at basic biology. Because isolated cells are interesting because they are subject to autocrine signaling only, meaning we are actually probing how these cells are secreting and responding to their own signals. And I'm going to show you how that can lead us to some interesting insights. Okay, so just a minute to show you what these data might look like. So if we stimulate macrophages with LPS and we analyze only the wells with single cells in them, if you see as we increase the concentration of LPS, you can see the high variability in both the fraction and the levels at which these macrophages are secreting things like TNF and IL-10 and IL-6. And so a few years ago, we published a study, and this first study was done in a macrophage cell line, U937s, where we showed that, remember I said TNF is one of the things secreted that goes on to affect other cells. What we showed is that the fraction of the top 5% of individual cells secreting TNF are actually secreting much higher than average levels, and in fact account for more than half of all the TNF in the device and also in the plate. We can also see these cells in the plate at the time. And so what we think then is going on is those sort of super secretors or precocious cells are basically secreting a bunch of TNF that then goes out to signal to its neighboring cells. And this can lead to, the cascade that I was talking about. But now the insight is that not every cell is participating equally in this. Some of the cells are participating more than others, and they're going on to cause other cells to secrete more things, other macrophages in the population. What's neat about these data, I think, particularly for the all the computational systems people in this audience, is that there is a ton of information encoded in these single cell secretion data. So if we take all these different wells with all the different combinations of cytokines that they have, and we use Bayesian network modeling approach where we can do directed acyclic graphs, we can sort of infer these from the data. We can actually recreate, just by looking at the data, this complicated paracrine network graph, where I hope you will appreciate that TNF is there at the sort of close to the top of the graph and is connected to all of these other cytokines. And if we block TNF, we will see that we affect pretty much all of the chemokines and cytokines downstream in the graph, but we don't affect CCL2 and CCL3, which are upstream in this graph. I should point out that the arrows in some cases indicate a direct maybe stimulation, in other cases might just indicate more of a temporal thing. So there's a lot to unpack in these graphs, but I just want to point out that at least some of the structure of the cascade is there. You can also see that IL-10 is there connected to TNF. And the other thing I mentioned, interferon beta and IL-27, these also form a little subnetwork that's connected to IL-10. And I also want to mention that all of this work in this first part I'm going to show you was done by Amanda Alexander, a graduate student who just graduated from my lab. Okay. So we wanted to sort of explore this IL-10 part of this regulation, though. This is acyclic graph, so we can't actually see the IL-10 feeding back to stop this response. So here I just want to provide some data to convince you that IL-10 really is a negative regulator. If we add an antibody to the IL-10 receptor, we will see that secretion of TNF and IL-6 in the green goes up in response to LPS. And if we add recombinant IL-10 and we sort of force the system to be negatively regulated, you will see that both of those go down. You can also see this over time and in the dose response. So over time, TNF rises first and IL-10 follows. And in the dose response, we see that TNF is activated at lower doses, but as the doses increase, IL-10 is activated more and and TNF goes down, and we would infer this is because of negative regulation of IL-10. Okay. So this leads to sort of an emerging picture where we have a heterogeneous collective mechanism for inflammatory activation and resolution, where LPS causes some cells to cross a threshold of secretion, and we have these TNF high secretors. And then they go out and mediate quorum sensing to activate more cells. And again, I'm talking about a community of macrophages on a plate, essentially, but this could also happen in vivo. And then finally, those high secretors, we think, end up secreting IL-10, which helps to resolve the response. And this isn't just us. I've listed some other references there that have also looked into this sort of quorum sensing mechanism. But what I want to explore now is whether that's really the complete picture. That's what Amanda wanted to ask as part of her thesis project. And so we used our microwell device to actually try to perturb negative feedback in this system. So we took the single cell suspension, we put it on our device with LPS alone or in combination with antibodies to the IL-10 receptor or recombinant IL-10. And then we visualized the multiplex data using FATE, which is a visualization algorithm that really tries to emphasize some of the global changes in the data, more of a manifold visualization of the data. And what you can see, I'm showing you the cell density on 10, 100, and increasing doses of LPS, essentially. You can see that cells are kind of moving into an axis along the x-axis. And what we think that is a pro-inflammatory axis of activation. And I call it that because if you color the intensity of secretion of the underlying factors like TNF, CXCL1, and IL-6, you can see that these are all increasing as we move along FATE1 axis. However, what I want you to note is that IL-10 and interferon beta actually are sort of more evenly produced along this axis. So if IL-10 was really happening only in response to the highest secreting cells, we would expect to see it almost entirely along the far right side of the axis. But instead we see it kind of throughout, including cells that are secreting quite low levels of other things. And so then if we perturb IL-10 and we block signaling, we actually see that kind of consistent with this, cells in the low end of the axis, the far left, actually start to move into that middle part. And on the bottom here, what I have is a kernel density estimate of the cells along the FATE1 axis. What I want you to appreciate is, although it's somewhat subtle, the blue, the lighter blue, you can see that when we block IL-10, we're actually releasing cells from a low to sort of a mid tier of secretion, in addition to actually bringing cells from the mid to the higher tier. And then if we add a recombinant IL-10, we force everything down to a lower level. And so essentially, just to restate this, what we're saying is that when we block IL-10, we are not actually just reining in high level secretion, but in fact, we're increasing the fraction of cells that respond. And here I'm showing that in terms of the fraction of cells that produce TNF. Whereas the average secretion actually goes down because we're recruiting all these new cells into the pro-inflammatory cytokine axis, essentially. So why did we think this is important? Well, a couple years ago, actually, there was a paper, a really beautiful paper from Ron Germain's lab by Rachel Gottschalk, showing that the LPS TLR4 mediated response has a strong threshold component. So one of the things that's always been kind of a mystery is that, macrophages need to discern between homeostatic levels of microbes and pathogenic levels. And so one could ask, how did they discern that? And this paper shows that in some ways it's dose, and that there's a strong threshold that's imposed by MAP kinase activity. And you can see this in the data where you see sort of a strong dose response. And so we tested whether IL-10 might also be contributing to that threshold. That's what our data would suggest. And in fact, that's more or less what we see. So if you look, we can also see this strong increase. If you look at the black dotted lines, you can see that as you increase LPS dose, we see a very strong increase in TNF, IL-6, and CCL5 secretion. And if we block IL-10, we actually see that we can move that threshold down lower. And we've looked, and this isn't affecting MAPK. This seems to be a separate mechanism in terms of impacting this threshold. And also I want to point out, this is happening at very low levels of IL-10. So I'm superimposing here the curve of IL-10 secretion in terms of percent of the max response. And it's at very low levels, we're actually seeing this activity taking place. And so finally, we wanted to test the role of interferon beta, because I mentioned to you that interferon beta is also known to participate in this, in activation of IL-10. And so here I'm showing you again, it's a different plate plot because it's a different set of secretion data, but you will see that we have this axis of activation. And when we block TNF, this is sort of a control, we can see that absolutely we see cells move out of the axis of activation, showing that TNF is definitely a positive regulator. However, if we block IL-10 signaling, or we block interferon beta signaling by blocking IFNAR, its receptor, we see the opposite movement, where cells now move from the low part of into the axis of activation, suggesting that interferon beta is primarily mediating this strong negative sort of threshold component of IL-10. And if we go back and do the same population level experiment where we look at the threshold change in TNF secretion across LPS doses, we can see that if we block IFNAR with an antibody, we see pretty much the same change that we see with IL-10. And so overall, I think this just contributes to a slightly more complicated picture of maybe what we think of as this emergent collective mechanism, where cells, although TNF is still there to kind of implement a positive feedback, we also see that interferon beta is there to mediate even very early in the response, sort of an IL-10 negative feedback that's keeping certain cells from crossing this threshold of activation. And then eventually we still think TNF probably participates in having IL-10 help to resolve the response. And so essentially there's sort of two roles of IL-10 in this process. And given that IL-10 has had a lot of, trying to perturb IL-10 in the clinic produces a lot of confounding results, we think that perhaps the fact that IL-10 is having these somewhat quite divergent roles and how it's regulating the response could be something to think about. Okay, so then for the second part of my talk, I think I have a few minutes left. I want to talk to you about another question. So let me pose it here. If we have these cells and we know that we treat them with LPS, and some of them seem to be, let's for the sake of argument say, poised to respond by making high levels of TNF, you might ask yourself, well, then are the other cells may be poised to respond to something else that macrophages need to respond to in their environment? And so that's the question we asked. How do macrophages respond with specificity, but also flexibility when exposed to conflicting environmental stimuli? And so here I'm going to raise this slightly controversial subject in the innate immune world, which is the M1 and M2 polarization of macrophages. This comes up a lot in disease, cancer, different, it's a framework that we commonly use to think about macrophage functionality. Now, I think we all know it's oversimplified and that it's probably extremes on this spectrum. But the truth is that macrophages are often exposed to conflicting polarization stimuli in vivo. And so we might ask ourselves, what happens at the individual cell level when a macrophage is faced with these two opposing cues? The other thing to tell you is that in the population, we know that there is significant cross-regulation between the gene programs of the M1, which is the in red here, which is the one that's activated by LPS and is more of a pro-inflammatory phenotype, and the M2 program, which is shown in green here, which is activated by IL-4, a Th2 cytokine, and leads to this more anti-inflammatory or resolving response. And so another graduate student who graduated a year ago, Andres Munoz Rojas, asked this question: if we treat cells with LPS or IL-4 or the combination, the combination of the two, will we get mixed responders completely, or will we get some cells actually choosing, quote, to respond to one of the different stimuli? And so first we had to, these are complicated programs, they have slightly different timing, so I'm just going to show you here that the IL-4 program, which is in blue, and I'm showing ARG1 and Chi3, which are two IL-4 gene, kind of canonical genes. You can see that these are rising over the course of 8 hours, whereas IL-6 and IL-12 and TNF, you can see these kind of peak and then go down between 4 and 6 hours. And so we decided to do 6 hours as our time point. I also use this graph to show you that there is strong inhibition when you add the two together. So you can see that ARG1 goes down significantly when LPS is added in green, and IL-6 goes down significantly also, you can see in green with the combination. But then something like TNF actually does not. So that, there's variability. Okay, so then we used single cell RNA sequencing to measure this at 6 hours. We used 10S genomics. Here I'm just showing you a snapshot of the program. And again, I want to acknowledge that the LPS plus interferon program is much stronger in the sense that you get over 1,000 genes upregulated as opposed to IL-4 where you get about 380. But I also want to, I get asked this a lot. We did look and we could show that almost all of the cells were responding to both of the cues, and we looked at that at the signaling level. We could see that the STAT1 and STAT6 phosphorylation downstream of each of these receptors, apparently you can see in red here, was more or less activated in all of the cells. But that was almost unnecessary because when we did the UMAP plot to look at how these cells look in sort of a global transcriptional state space, we can see that the green cells, which are the co-stimulated cells, are absolutely occupying a different global state than either the LPS program or the IL-4 program alone. I think it's pretty clear. Almost every cell shifts, suggesting that almost every cell has been affected by both cues. This was actually not that interesting to me. I think that sort of makes sense because they're all, in some ways, both programs are going to upregulate lots of different things. What I was more interested in is what things might not be the same. And so you can see that there is a lot of heterogeneity in those green cells in terms of, for example, some canonical things like ARG1 and IL-12. And so we wanted to explore that a little bit further. And so we decided to focus on, I already told you that some things are negatively regulated, like LPS negatively regulates IL-4 and vice versa. So we focused on the genes that had that property because our thought was, well, maybe that is a way the cell is trying to impose some order on the system, for example. And so we found then 87 genes that are upregulated in response to LPS, but downregulated in response to co-stim. That's what I'm showing you there in the middle. And then on the right, there's about 200 that were upregulated with IL-4, but downregulated with co-stim. And so those are the ones we focused on next. And again, some of our favorite members are part of that. I'm showing you here some plots of IL-12 on the top and IL-6, as well as ARG1 on the bottom. Alana Kelsey, a postdoc in the lab, helped with this data. And what I want you to appreciate here is already we can see from the single cell data some pretty interesting things. So in the population RT-qPCR data, we see that something like IL-12 in yellow is upregulated, and then in green, when we add IL-4, it's definitely inhibited. But if you look at the single cell sequencing data, you can see that the tail of the cells producing IL-12, the fraction goes down, but the tail is exactly the same, suggesting that these cells are some cells are strongly producing IL-12 as if they've never seen IL-4. And you can see a similar thing for ARG1, right? Suggesting that there's a huge increase in noise potentially in terms of how cells are expressing these. So another thing I wanted to point out is sort of exciting to us is that if you look for enrichment in the pathways that are among these negatively regulated genes, for the LPS genes that are negatively regulated by IL-4, we actually saw an enrichment in communication between innate and adaptive immune cells. And given our interest in signaling and sort of extracellular paracrine signaling, we thought this was of interest. Okay, but really what we want to do is we want to find these genes, these things that we think might be specializing or basically responding to kind of one or the other stimuli. So how do we do that? Well, we reasoned that we can't really look for anti-correlation. What we're looking for is basically the probability of expressing A would decrease the probability of expressing B and vice versa. So we realized that the odds ratio would be a good way of doing that. And so we looked among the cells that were co-stimulated, we looked for genes that had, if the log of the odds ratio would be negative, or essentially the odds ratio is less than 1, meaning if you express A, you're less likely to express B. And satisfyingly, we found that those are mostly found between genes that were IL-4 versus LPS genes. And kind of interestingly, it's not that many. There were only, there's a small subset of cells that have significant odds ratios. But when we looked at them, and you can see here on the right, there were some really interesting candidates. So for example, we can see that IL-6 and IL-12 do in fact have significantly negative odds ratios with ARG1 and Chi303. And potentially even more exciting, their transcription factors also show some negative odds ratios with transcription factors from the other targets, suggesting that through this method and this just sort of gathering these data from these snapshots, we can start a little bit of a mechanism and the regulatory network emerges. So what does this look like? Well, I'm showing you now plots here, and it really does look like orthogonal gene expression. So if you look at the top two, we have ARG1 on the y-axis and IL-6 and IL-12 on the x-axis, and you can see that they are very specifically expressed in response to LPS and IL-4. And if you look on the far right, you will see that there are not many cells in the upper right quadrant, meaning that they either express one or the other. And I just want to point out to you that is not the same for TNF. TNF is expressed in cells, whether they produce ARG, and cells producing ARG just fine. So this is sort of a special property of certain genes. And then just to show you the transcription factors are also orthogonally regulated. It's less striking, but as I mentioned, we think that there's literature evidence to support some sort of transcription factor network that we're now trying to test experimentally. Okay, so the last thing I want to leave you with is whether this matters at all, okay? So what I've just showed you is transcription at 6 hours. And you might ask yourself, well, does that actually matter functionally for cells down the line? And so ARG1 is not secreted, but Chi303 is. And Chi303 also shows this orthogonal expression pattern with IL-6 and IL-12, which are secreted factors, again, that I mentioned. And importantly, these are secreted factors that communicate with T cells. They are T cell polarizing cytokines. And so what we wanted to do, we have our single cell secretion device. And so we could actually ask, at 48 hours, do we see secretion that's orthogonally regulated between these two subsets? And so that's what we did. We put the cells in the device. We waited 48 hours after co-stimulation. And what I hope you'll appreciate is that we do in fact see that 48 hours later, we have cells, if you see on the far right there, cells that are mostly either secreting Chi or secreting IL-12 or IL-6, but generally there's many fewer cells secreting both, consistent with the transcript data at 6 hours, suggesting that this is actually a durable response of the cells. And again, we do not see the same thing with TNF and CCL5. So with that, I just want to end and say, I think the last point is the one I want to emphasize the most, that we actually think that increasing functional diversity in the population might be a strategy macrophages use to respond to conflicting environmental cues. And again, this is not for every program. This is for some programs, for example, potentially the Th1 polarizing cytokine program. And so we think that this might also be a way to sort of think more specifically about what we mean when we're talking about how cells are diverging on sort of an M1 or an M2 axis. Maybe it's important to define exactly what we mean and which functions that we are talking about. So with that, I want to thank my lab and the funding sources. I tried to thank the people who did most of the work along the way, and I'm happy to take questions.
All right, wonderful. Thank you, Kathryn. I gave you a round of applause. All right, cool. Are there any questions for Kathryn? Please type it and I'll get started. So I'm always impressed by this technology that you are using for single cell proteomics. So I'm just curious, if you were to generate from the same sample single cell RNA-seq data, obviously it's not going to be same single cells, but do you think we can capture the single cell secretion or infer the single cell secretion from single cell RNA-seq?
Yeah, so we're really interested in that. I will say, so we've started to do this a lot in the tumor microenvironment. We've been gathering macrophages and other immune cells from the tumor microenvironment. And you know, so far, generally, we see rough correlation between transcription and secretion. What we're really interested in trying to do is to see if we could go backwards and we could actually sort of, from secretion data, kind of try to identify the subset of cells that might have the transcripts, and then underlying that, see if we could find markers of, I think this is what you're getting at, like, could we actually sort of predict, given a certain transcriptional signature, what their functional secretion might look like? I think that's what you're asking, and I think it's possible. It's something we're working on, but we haven't finished yet.
All right, keep us posted. And one other quick question.
Can I ask a question?
Go ahead, Farhad.
Thanks, Kathryn. A great talk. So in I think CD4 T cell effector differentiation or memory differentiation, there is some work that's showing that we like to believe that the naive cells are identical, all identical, right? And very homogeneous given many other cell types. This is really nice. But it seems like if you do single cell attack-seq, then you may see signatures of things that may go towards Th1 versus Th2 versus Th7. So is it something that you consider in your macrophage polarization as well, or?
Yes, absolutely. We're really interested in trying. In fact, we're getting ready to try single cell attack-seq. For a few other reasons, data I didn't show is that another interesting aspect of the IL-4 versus LPS response is that the IL-4 response appears to be sort of quick, and that you don't need a very long stimuli to get a long stimulus duration to get the response, but you only ever get sort of like 40% of the cells responding, as opposed to LPS, where like it sort of keeps increasing over time, the fraction you can get. And so we think that something like attack-seq would really help us start to answer how that's happening. Yeah.